So our next speaker, as you can see, is Norma Paniego. I invite you, Norma, to be there, prepared for the talk. And I will tell you, Norma Paniego is a principal researcher of CONICET and also researcher for INTA, which is the National Institute for Agronomical Technology, which is a very important institution in our country. And she works at the Molecular Biology Institute in INTA, also a member of CONICET. And Dr. Paniego leads different research lines aimed at the study of crop complex traits, combining cutting-edge genomic techniques, bioinformatics, and molecular breeding. Her research also includes the use of genomic data in genetically diversity studies of cultivated species, important for food security and ecosystem services, for its use in breeding and conservation. Her activity as coordination and also sometimes as co-coordinator of institutional projects and national and international consortia has resulted in the generation of resources and capacity building in genomics and bioinformatics in our country. I can tell you, she's also a reference in all those projects and techniques. INTA is a very important institution in our country. As you know, we are highly producers of bovines and crops, and we are a country that needs a lot this type of help from these people working at INTA. So I leave you with her. I stop sharing my computer. And Norma, are you there? Can you open your camera?
Yes. Yeah. Can you see me?
Not yet.
Yeah.
Hi, nice to see you. Norma. It's a long time since I've seen you.
Yeah.
Nice
Thank
to hear
you very much for the invitation. It's a pleasure to be here and an honor to be invited to this first Women in Bioinformatics Congress. So,
glad to see you. So I leave you. You have 30 minutes. And
okay, I share the screen. Well, I start with talk in the coming 20 minutes. I will try to tell you about our experience integrating bioinformatics to crop breeding. So the talk has this content. We will briefly introduce the Institute as a research group. I will give an overview of the moment we enter in the field of the genomic and bioinformatics. I will tell some progress in research related to molecular breeding, mainly for disease resistance in sunflower, and also talk about bioinformatics tools, research progress, and opportunity for training in bioinformatics. Well, this is, we belong to the Institute of Agrobiotechnology and Molecular Biology, which is a double dependency unit from INTA CONICET. So we work at the Research Center for Veterinary and Agronomic Science. It's located in Burlingame, Provincia de Buenos Aires. And the group was formed in 2000. We started working with Ruth Hen in sunflower genomics. Paula Fernandez is working in functional genomics, Veronica Lia in population genomics, Máximo Rivarola in bioinformatics. There are young researchers, Emiliano Benguerrero working in plant microbiome, the data effects that can contribute with defenses, plant defenses. Monica Paz working with Mariana Conte in tilling population, induced mutation, and started with target mutation. Carla Filippi working in association mapping and genetic diversity. Juan Montecchia, working in association and biparental mapping for QTL discovery. Sergio Gonzalez, working in bioinformatics, together with Massimo Rivarola. He is in charge of the computing infrastructure, and his research line has to do with the optimization of the assembly of RNA-seq reads. Pablo Aguilera works in host pathogen interaction in the pathosystem Verticillium sunflower. Salvador Nicosia works with Paula Fernandez in senescent process in the molecular characterization of senescent process. Andres Ribone works with Maximo in the reconstruction of gene networks. Matias Dominguez is a breeder working in the development of a multiparental mapping population. Nicholas Haynes works in drought resistant in association mapping population, and Denis Colombo works in the pathosystem of the Aporte Lianti and sunflower, working mainly in the pathogens. Well, just to put in context, when we start with the bioinformatics and genomic area, it was at the late of the 90s in the what is in the timeline for plant breeding is the 3.1 step. That at this time INTA already has been adopted a molecular marker, anonymous molecular marker. Jorge Bukowski was back from his postdoc from United States, working with PCR marker and promote the adoption of this kind of technology for breeding and for control. So he established a project together with the National Institute for Seed and seed companies to utilize this type of marker, are also developed in the case of sunflower. That was a small crop at international level that need that kind of tools to be developed. So we start in this moment, it was 1997, with this project, and immediately we understand that we need some computational tools to work with the sequence that we were producing by Sanger sequencing. In 2000, with Ruth and Paula, started to produce another kind of data, ESTs, just to complement SSR from a tool that comes from the functional part of the genome. From that time to now, our research line progressed to more sophisticated goals, and our publication now target the genetic basis for resistant fungal disease resistant, reconstruction of gene network associated with senescence, or utilizing data, big data available in public repositories to understand the way the international breeding program utilized genetic resources for sunflower breeding. So I include this slide just to illustrate the title of the talk, because, well, I want to transmit which was the impact of producing genomic data associated with breeding program in the progress of the research group. We start producing SSR microsatellite, allow us to work with breeders characterizing core collections and biparental population, and developed different research lines associated with the study of genetic diversity, of population structure, of QTLs, focusing in disease resistance and also abiotic stress resistance tolerance. Then we produce ESTs and study specific inbred lines that has contrast behavior to the process we were studying, and identify some candidate gene to complement the QTL studies. And then in 2005, we start producing SNPs, and we approach population association mapping population, and explore new breeding strategies for stress resistance. And at the time we were developing new data, we also developed or adopt bioinformatic tools for variant calling, for functional annotation, for genome and transcriptome reconstruction, and for data management and storage. And these capacities allow us also to offer training, not only in bioinformatics, but also in genomics. And we have all the opportunity to get some equipment for sequencing, for multiplexing, and this results in services based in sequencing. We get also some high-performance computing servers to use in crop studies, not only sunflower, but forest crops, animal genomic animal studies, and microbiology genomics. So this is the impact we get in our research line and in the institute. Coming back to sunflower, and to illustrate the adoption of the SSR and the SNPs in the study of the biparental mapping population and association mapping population in a complex traits that were that has impact in the sunflower production in Argentina. Jeremias Zubricki and Carla Filippi, working with biparental mapping and association mapping, and implementing this kind of resources, associate the genotype data with phenotype data collected during different 4 or 5 field trials, measuring different variables associated with the disease, identify different QTL or genomic region associated with the response defense, spread across, in the case of the biparental population, almost all the chromosome, and it was less distributed in the case of the association mapping, and analyzing the phenotypic data collected during all the field trial. Carla also identified those accessions that stack the major amount of alleles that positive impact the defense, the reaction defense of sunflower plant to the Sclerotinia attacks. So this kind of information is very valuable for the breeders, and give us a support for new research lines. Another way to identify candidate gene associated with a different response is through transcript analysis. In this case, Monica Fast analyzed data coming from free accession that has contrastive behavior to the disease, and she identified different a set of a small collection of differential expressed genes, and some of these genes were colocalized with QTL associated with the sunflower head rot by biparental mapping association. So we get in some way a validation of different genomic region that has a role in expressing that response. Another interesting thing that she found in this small sub-collection of candidate gene, it was that the annotation was non-coding RNA molecules that has very clear differential response in all the genotypes. So this result opened new interest in the group to go deeper in the response of sunflower to this complex disease. 7 years ago, we we work with new generation sequencing, and this technology allows us to develop new genotyping techniques. So we developed one target genotyping technique based on African sequencing applied by Monica to study teething population to identify new mutant associated with disease responses and to validate candidate gene we found by mapping QTLs, for example. In the other place, Carla Filippi with Natalia Aguirre developed DDRAT, genotyping by sequence methodology that focus on genome-wide SNP distribution. This technology has the limitation that accumulate missing that data. Gabriela Merino helped us to develop a strategy to impute data and get a complete matrix for further analysis. Juan Montecchia implement this DDRAT technology in the biparental population and use also the association mapping to study another important fungal disease in sunflower, which is verticillium wilt. In this case, he found 33 genomic regions associated with resistance and 9 regions in the biparental population, and analyzing in both results, found that some QTLs in chromosome 5, 10, and 14 co-localized in both populations, which gave also some power to use those regions in breeding. And he also recognized in chromosome 10 that hold a lot of defense trait, not only for verticillium, but also for sclerotinia, a region that explain a big part of the phenotypic variability observed in the population.
Sorry, Norma. Sorry, you have 5 minutes left.
Okay. Well, I'm integrating the result obtained for sclerotinia and verticillium. We can identify different accessions that accumulate beneficial allele, and we start the development of a multiparental population that is also very, very power resource for breeders. Carla had the opportunity to exploit data coming from the international sequencing project from Sunflower to test, well, to see how each this population contribute to the breeding at international level. She found, she called different ethnic data set for these 3 collections that are the international resources that Sunflower has. And well, she recognized almost 12 shared SNPs that allow the study of genetic diversity, population structure, and linkage disequilibrium. And well, the conclusion of her work is that even when the collection, for example, the group in blue comprised most of the accession from INTA, and this comprised the accession from USDA, but some of the group mixed accession from the 3 collections. So there are some kind of homogenization in the 3 collection accession that demand in the future the incorporate new genetic variability to improve the power of the breeding program at international level. I would want to introduce also some bioinformatic resources developed in a group. One is ATGT, a web-based application developed by Sergio. This platform allows us to storage and also explore and visualize different data sets produced in INTA. And I finally I want to comment some of the Sergio research interest that has to do with the optimization of the local assembly. As the previous speaker talked, it was very difficult to get a good assembly with a short read. So the experimental design for Sergio research is working with sequence grid simulated from Illumina technology, and he tests the performance of the 5 or 6 public assembler, and test different metrics to check the quality of the result of these assemblers. Well, he has a very good result in terms of how is the performance of different assemblers in front of the biological complexity present by the alternative splicing, the number of family genes, or the a dynamic range expression of the transcript, and also how is the impact of the side fragment in the case of the technology implemented. Well, I also want to comment about Cabana with this consortia for capacity building for bioinformatics in Latin America. I recommend the audience to have a look to this platform because there are a lot of opportunity for capacity. Now all are directed to e-learning, and these are the possibilities for e-learning in Cabana site, but it's very dynamic the way this site is updated. So you have to be updated with that information. And I want also to comment that tomorrow Natalia and Carla Filippi will give a course about CBS data analysis. This course is the first time that is completely virtual, and maybe will be available for those interested that were not will be not present tomorrow to assist in the future. Well, and that's all. Thank you very much, and sorry for being just five minutes more.
No, don't worry. Thank you very much, Norma. We have few minutes for a question. Does anybody has a question? Very few minutes. I have a comment more than a question. It's amazing how things were changing during the time, and in a short period of time. Data change, the technology, the way we have to analyze the data. So it's really challenging to be all the time at the cutting edge of the technology.
Yes, yes, yeah. That is also very, very exciting. The possibilities of the new technologies, as the last speaker talked. Now the third generation sequencing and the possibility to have a long reads, open, completely different landscape, especially in our case that we are looking all the time for variables. The possibility to analyze structural variant is very amazing. Also, the possibility to reconstruct or identify isoform, that is the problem with transcriptome assembly nowadays with Illumina reads.
So nice. It's really amazing how INTA was changing and your group was changing, mutating all the time with the new data and technology. So thank you very much, Norma.